So this is an application of phasors, in particular hyperspectral imaging and phasors. And this was a collaboration with a group at the University of California in Irvine when I was doing the postdoc, and it continued after I came back to Uruguay and stabilized our group here. Because this was interesting. There was a person in the Department of Developmental Biology and Cell Biology, one floor on top of the floor where we were in the California, interested in understand how is the dynamics of the water within the zebrafish lens. And we were doing experiment with Andres Camay, because he was visiting me. We were doing some experiment with the zebrafish, interested in another thing. And this person, Leonel Malacrida, asked us if it was true that we were doing measurement of water. And she was completely interested in the water inside this lens. So this was the beginning of this collaboration. But I have been working with the dynamics of the water for a long time. And at the end of my talk, I will come back to this, and I will try to also make a stop for my great friend, colleague Luis Bagatoli, who passed recently, as I mentioned before. But he influenced me a lot in the understanding of the biophysics of water within the cells. And during my PhD thesis, I was interested in the organization of the lamellar bodies, as I mentioned before. And then when I moved to California, he introduced me with the ACDAN, who is a small brother of the Laurdan, using this case. Laurdan was for membrane, ACDAN is for intracellular water dynamics. And when I was working in California, I spent a lot of time trying to understand the dynamics of water inside the nucleus, in particular the different kind of chromatin. And this was never published because I never have the time to write the paper. But basically, we study a lot about how is the dynamic of the chromatin in different situations, and there is a lot of work done in that case. And Luis was very influenced in my research. I don't need to introduce much about these dyes, but as I mentioned before, Gregorio, Weber, and Ferris introduced this series of dyes, which are called dimethylaminonaphthalen. And if you change the R group here, you can give different affinities for the structure. And with the methylene group, it's called ACDAN. It doesn't have any affinity for membranes, or it has a very poor affinity for membranes. It's outside the membrane. And it's a wonderful tool that we use it a lot to measure the dynamic of water within the cell. For our purpose of this work, we were using ACDAN in spectral imaging. And when ACDAN moves for an environment which you have poor water or confined water, it moves from blue to the green emission. And we were using, of course, spectral phasor to measure this shift. And when I met Irene was postdoc at the Tom Schilling lab, and also the James Hall in Irvine, California, and she was interested in the lens biology because, well, lenses are fundamental for the vision. So these are our our biological lens to focus the light in our retina. And it requires really transparent condition in order to form a proper image. And if you have any problem in the lens, like cataract, you will produce blindness. And this is one of the major situations in the world where you have, blindness by cataract. As a model, Irene was working with zebrafish because, well, for those of you who know this little animal, this is very nice because it has a rapid development. It has a small size, which is completely compatible with the imaging tools. It's transparent, and this is very important. And it's very friendly to do genetical manipulations. So Irene is an expert in this. And we started this collaboration probably in 2017 or something like that. And it continued until the last year when we published finally this paper. Meanwhile, I was coming back to Uruguay. Belen was going to the LFD, and she took my place and helping Irene in all the imaging. So Belen also played a big role in this work. But let me tell you a little bit more about what was the interest of Irene. Irene was studying these proteins, aquaporin 0, in the lens. These are specific aquaporins for the lens. And these are duplicated in the zebrafish because the teleos duplication. And it has two versions, the aquaporin 0A and B. They are both required for the proper transfer of water within the lens and the proper homeostasis of the water in the lens. Mutation of the aquaporin 0 produce this very aberrant organization in the lens. And Irene was already studying the structural organization. These are, imaging of phalloidine and DAPI. As you can see that when she mute the aquaporin 0A, you screwed up the lens. With the aquaporin B, you screwed up, but not that much. But when you knock down the both, the lens is really completely aberrant. And the transparency of the lens go from this situation to this really bad situation where you cannot see through the lens. So with that, we start a collaboration, and another person take place in this project, and this person was Alex Valmichana. I don't need to introduce all of you. You know Alex. Alan was the computing person of the project. And the problem that we have for analysis and study of this project is, well, Irene was really an expert mounting the animal, but the animal need to be facing the objective, right? And we have here a chamber, and this is the glass. And the animal was positioned, but it was impossible to reproduce one to another, exactly the same position. And we wanted to understand the water dynamics in the cross-sectional at the center and the different regions in the lens. The lens is not round, it's like a walnut, it's like elongated in the bottom. So it has a weird thing. And what we did is, well, first of all, we acquired 2 photon images in 32 channels in this size 710. And then in this stack, you can have spectra for one on another. And Alex performed a routine in MATLAB to do the transformation. And then we color the spectral shift with this kind of scheme that select the different angles, so the different modulation, which is related with the change in the center of mass of the spectrum. But the problem of analyzing and comparing different lenses is that the center sometimes was here, sometimes was a little bit apart and apart. And we wanted to do really statistical analysis. We analyzed it like almost 100 of animal per condition. And to do this by hand was impossible. And I like this amazing algorithm in which by the expert, Irene, she select the center and then 4 reference points to isolate where is the perimeter. And then Alex was able to transform this circular shape of the lens in this flat shape of the lens. And this is as you can see, this is the center of the lens, and this is the perimeter of the lens, and this is the cross-sectional of the lens. So he extrapolated all the measurement of the dipolar relaxation of the lens in all the angles, and then we were able to analyze the distribution of water across the different layers of the lens for all the lenses we wanted to do. And this was impossible to do by hand. Only because we have Alex, we were able to do it. So the first thing that we need to test is, well, how was the response of ACDAN to the crowding, right? And crowding means by the increased amount of proteins, which compromise basically the availability of free water. And the most important protein in the lens is the crystalline. And you see here, starting from here with ACDAN in water, as much as you increase crystalline, the position of the phasor move toward to the 1,0, which is the blue, right? It's getting more and more blue, the spectra, which means lower and lower dipolar relaxation or higher and higher molecular crowding. And then we want to study how was the development of the lens across the time. And there is interesting point here. At the early stages, the lens cells has all the organelles. But over the time, they lost the intracellular organelle, the nucleus, the mitochondria, etc. And these cells are basically staying there without this organelle, and it's really important for the hemostasis to bring new nutrients and move the waste with these changes in the osmotic pressure, etc. So these are the images, as you can see here. At the beginning, we have like a very heterogeneous distribution of dipolar relaxation. Red here means high dipolar relaxation, and blue or purple means more compact or more organized. And you see that over the time, the interior, which is known as the core of the lens, start to get more and more crowded. And the dipolar relaxation is pulled a little bit to the equatorial region of the lens. And this is qualitative analysis, but consider the algorithm that Alex developed. We were able to analyze, as I mentioned, hundreds of animals and different stages. And this is the distribution of the length diameter, which is a way of measuring how old is the lens or how old is the animal, and the dipolar relaxation. As you can see here, this line, black line, is from the beginning. The high dipolar relaxation is in the center of the lens, and then moving over the time as it's going to some point in the middle of the equatorial plane, as is described by this sigmoid here, as well as here. So first of all, we define that at the day 4 to 6, the lens got mature, and in this situation, the lens is capable of seeing. So it's capable to focus the light on the retina in the proper condition. And these are the correlation measurement of how changed the interior of the dipolar relaxation in the core or in the inner cortex. And we can measure this and do some statistical analysis without any problem. Then we were interested in understand what happened in the anterior and the posterior plane, because this is where these cells stick together. They call suture, like where they close all the cells in the superior and in the bottom. And there was a different behavior in the top, in the suture, and in the bottom. In the bottom, there was like a biphasic. It was like a low, high, and low, and in the other was more or less following the same that we saw in the center. So basically, the tightening of the anterior suture is developed, during the maturation of the lens, and in the posterior suture, we see this increase after 4 days, but then it got tight. And also, if you compare anterior and posterior, it is a difference between the water dynamics. The second important question we wanted to answer was in the native situation is, how was the organization of water across the lens? And what we did is, well, we got CT stack, and this is a CT stack, right? And then we did two analyses. One was, okay, we will analyze with these algorithms of Alex, what was the average, basically the distribution of dipolar relaxation along the lengths in the different CT stack, or he generate an extrapolation in a cone perspective along the whole thing. So he interpolate the points to measure the dipolar relaxation in the same point in a 360 analysis, but in the vertical position with one aim. The idea was to figure it out if the distribution of the water and the organization of the lens was cylindrical or was spherical. And as you can see here, in the conical slices, you see that this is flat. So the relaxation is always have a modulation, because in the center is flat, is low, in the other part is high and then low, and in the other is the other way around. So when you got from bottom to top, you got always the same, because in average, the dipolar relaxation is the same. So we demonstrate the distribution was spherical rather than cylindrical. And then we jumped into the problem, which was to analyze what happened with the aquaporins. And this is the transmission that was published before with the NIN analysis. And this is what happened with the dipolar relaxation in this situation. You see that they are immediately apparent differently. The most worst situation is in the aquaporin double mutant, where you have really high dipolar relaxation. And this explains this aberration in this refraction we see here. But we measure this using this analysis, and we identify that the most important difference happen in the inner and the outer cortex of the interiors. And all these measurements were done at the stage of maturity of the lens. It was, I don't remember, it was 4 or 5 days. And this was completely significant in all the analysis. And I want to stress again, this was like 100 animals per condition. It was really an amazing. And we did the same also for the anterior and posterior. And we got that the dipolar relaxation is lower, in all the regions of the lens in the aquaporin 0A. And there is, of course, a disruption of the water activity in the single mutant as well in the double mutant, which was really, really worse, as you can see here in the image. And something that we know a priori was that the aquaporin B was more related to adhesive properties, while the aquaporin not. And this was also confirmed by this analysis of the water dipolar relaxation. When we analyze the anterior and posterior plane in the different thing, we also can measure, the quantities, and these quantities were significant different in all the different part of the lens. So to conclude this work, we provide new knowledge about the use of this ACDAN. It was the first time that ACDAN was used as a ruler of the water dipolar relaxation on an in vivo animal, right? That can give you really a readout of water dipolar relaxation or molecular crowding, in other words. We contribute to understanding, the homeostasis of water in the lens and how this is important for the functionality. For me, from the biophysics perspective, it's amazing that a biophysics property can explain a physiological product, which is you are blind, you cannot see, no? And this is because molecular crowding, and this is because water dipolar relaxation at the end of the end. Then we understand what was the problem, and this is related with the swelling, because cells can get water in but not water out, because when you remove aquaporin B, is related to the transport and not adhesion. And we propose some model that Irene explained with the relation with the ions. Ions are very important. pH, and the different situation. So this was a fantastic collaboration with Irene and Alex and Belen, as well Enrico, Tom Schilling, and Shane Hall. And it was published the last year in Science Advance, and it's a really nice story that we are looking forward to continue, because there is a lot of questions. Finalize the day today and tribute basically honoring to Luis Fagatoli. I want to mention this because, well, Luis was very important for me, and unfortunately we lost him very, very soon this year by basically a disease related with cancer. And but Luis was very important for me because he introduced me with Laurdan and water dynamics, and we were working together before he passes. And I have to take care of this last paper of Luis using ACDAN to understand the interior of the cells. And somebody was asking me about the use of these dyes in plants. And this was this paper related. It was a new collaboration of Luis with some people in Córdoba, interested in understand the water dynamics in the leaf and in some other parts of the plant cells. And this was the final figure of this paper, where we figured out that there is a change in dipolar relaxation that depend on where you are in the plant, but also we discovered that there are some regions where they exist water as we want in our glass, but in some other are basically very confined organization of water. And I also want to bring Luis to this course because there are a lot of students, and through the Latin American bioimaging that you will learn more, because tomorrow where we finish with all the lectures, I want to talk a little bit about Latin American bioimaging. We generate a new prize in honor to Luis, which is an early career imaging scientist prize. And we will be announcing next year, the opportunity to apply for support students, for regional fellowship, for PhD and postdoc, travel, and to go and visit labs or core facilities that will enable to do better in their thesis. And this will be basically a call that will have an annual periodicity, will be selected by the executive committee of LABI, and it will be basically standing on the support of excellent scientific and critical thinking, as Luis always teaches. So I wanted to bring Luis because I think it will be an opportunity for all of you in the future to apply to this grant, and we will be very happy to keep his memory between of us.